All right, welcome back, guys. Now we're going to do our quick review for chemistry. I know most of you guys, I say most, you absolutely had to have taken some chemistry in order to be in this class. My opinion is that you should have taken some biology as well, but I know that it's not required for most of your programs, so it's a little bit unfortunate for me, but it is what it is. So this is our chemistry review, and this was chapter 2 in your text. Of course, you guys have already gone over the lecture, and you know already that this isn't going to cover everything in chapter 2 that you will be required to know. This is basically just going to be covering, be a quick rundown of the major concepts covered in chapter 2. So beginning, we know everything's made of atoms. Matter in general is made of atoms. Atoms consist of, in their nucleus, we have the protons and the neutrons. Those are the bigger molecules. They usually equal each other in number. And then on the outside, we have electrons that are flowing around. And oftentimes, usually, unless we're talking about things that are radioactive or affected by, radiation, usually your electrons are going to equal your protons, right? That balances out the charges. So the electrons have that negative charge, protons are the positive charge, and the neutrons have a neutral charge. And that's atoms. So most of you guys should be aware of that by now. When we get into talking about a lot of the stuff in microbiology, you absolutely 100% do need to understand your biochemistry. I know you thought you were going to take that class and let most of it go. This is probably going to get into a lot more detail than you ever wanted to get into in your class. They should probably call this biochem too. So just saying. So we're going to get a lot of microbiology, and I'm not trying to overwhelm you guys with this, but a lot of microbiology and what we're going to learn and talk about in and throughout the entire semester requires an understanding of organic chemistry and biochemistry. Biochemistry is part of organic chemistry. Okay. So if you did very poorly in that class, or you just threw that right out of your brain before you came to microbiology thinking that you were completely done with it, which would be, if you're going into healthcare, naive, but that's okay. You're going to use it a lot when it comes to the pharmaceutical aspects of things, right? So yeah, you need to go brush up on that if you haven't already gone through this chapter as a whole. I am just giving you a heads up because we are not letting this go after this chapter. Okay, this is going to carry throughout the entire semester. I'm going to be taking concepts from this chapter and even more complex concepts that you should have learned in your past chemistry classes that are going to be popping up quite a lot in microbiology, and really, literally, will be with us until the very, very, very end, the very last day. So, yeah, brush up if you haven't, okay? All right, the types of bonds in chemistry. The basic ones that I need you to know about for microbiology. Covalent bonds. Covalent bonds occur whenever atoms share their electrons with one another. So one is not taking it away from the other one. They are literally just sharing their electrons with one another. Anytime that you see any sort of a structure, like if we have a carbon and a hydrogen bound to one another, and you see it drawn with that line, that solid line, that is referring typically to a covalent bond. These atoms would be sharing their electrons with one another. Ionic bonds involve donating or transferring an electron from one atom to another atom. So if we have sodium chloride, which don't everybody answer at once, you know what this is, sodium chloride? Sodium chloride is table salt, right? So if we put this into water, we can dissociate these from one another. This is not a secret, right? So we put this into water, we can get sodium ions and chlorine ions, right? They came with one another in that solid form, putting them into water and dissolving them out, and now they're going to separate. Sodium in this case would have a positive charge and chlorine a negative. Why, though? Why do they have those charges? Well, because they had formed ionic bonds with one another, and chlorine stole an electron from sodium. Remember, electrons have a negative charge. So if chlorine stole that electron and kept it for itself, it's going to have a negative charge. It has more electrons than it's supposed to. Sodium, however, now has a positive charge. Okay, and that's because it lost its electron. So the charges in its atom aren't balanced out anymore. Why does it do this? Well, basically the reason that whenever you have ionic bonds like this occurring, the reason that this happens is because the electron orbitals. Now I know you remember over this, right? The number of electrons, hovering outside in the outer orbital of these atoms, they like to be like a certain nice even number. So like 8 on the very, very outside, or 2 on the inside, and then 8, and then 8, and so on and so forth. So they like to be balanced that way. And so they might give up an electron to something that, to something else to get to that steady electron orbit. Whereas chlorine might have like 7, and it just needs 1 more to be more stable. So now it has that negative charge because it has that extra electron, but its orbits are balanced. That's what happens with ionic bonds a lot of the time. Okay, so whenever we have something like carbon, which has 4 electrons on its outside, well, it's not totally unstable like that. But it can create 4 bonds on its outside, but it's not stealing everybody's electrons. It will share those electrons to keep itself stable. So now we can be happy together, sharing these electrons and not stealing. These other guys that form ionic bonds are happier when they steal, when they have the electron for themselves, because this other guy's happier when it gives it up. So that's the difference between the covalent bonds and the ionic bonds. Covalent bonds, we are sharing our electrons within those atoms. And we are in ionic bonds, literally donating or stealing electrons, right? That's what's going to transfer from one atom to another. The third type of bond that we are going to talk quite a lot about in microbiology will be the hydrogen bond. And essentially what happens in a hydrogen bond, so let's say we have, I don't know, a big old molecule of something, and let's just draw that molecule like this, it doesn't matter. And on it, we have an oxygen, and then it has a hydrogen bound to it. Okay, so I'm drawing these with steady lines, and so I want you to focus on this area here. So we've got these nice solid lines between these two, one line from the oxygen to the hydrogen here. This means that they're sharing electrons, right? Oxygen tends to be an electron hog. So the electrons are going to spend much more time on the oxygen side than they are on the hydrogen side, okay? So this happens no matter how big the molecule is. So let me draw you another molecule. So let's draw this one. We've got an oxygen, and in this case, oxygen likes to make 2 bonds. We've got 2 hydrogens bound to it. Does this look familiar to anybody? It should. This is water. This is H2O. This is H2O. It's a very important concept when we're talking about water being the dissolving, substance of all life. The most important solvent, which is a solvent, is any sort of liquid that other things get dissolved into, right? So the most important solvent in life is going to be water. And that's no secret, right? Well, you know that we're mostly water. Okay, so oxygen tends to be an electron hog. So it's going to have this negative charge to it because it is hogging the electrons from these poor little hydrogen atoms. And hydrogens, you guys, they've only got 1 electron. So when you start hogging that from them, they don't have a whole lot to give up there. Oxygen's going to hog those. It's going to give itself a little bit of a negative charge to it because it's hogging them so much. And the hydrogens, as a result, get a positive charge. This aspect of water is what makes life life. So now we've got positive hydrogens and negative oxygens. And these guys can interact with other things that are positively charged or negatively charged, or be repelled by things that don't want to have any charge or don't want to have anything to do with a charge. This affects how all molecules in life behave with one another. Is this. So anytime that you have, let me get an eraser. Hold on real quick. Need some space. Okay, so let's say we have another water molecule. So here's our one water molecule here. And here comes another one, because this is all water. So there's a whole bunch of water molecules next to each other, right? So we've got an oxygen with a couple of hydrogens. Don't growl, Ripley. Don't growling over. So again, slight negatives. We got these positives. So again, the oxygen that has that negative charge to it is going to be wanting to associate with that positive charge on that hydrogen, because positives and negatives are going to be attracted to one another. That's how a lot of this stuff works. So this right here, that association between that negative molecule, that negative atom, and that positive atom that have these charges due to hogging of these electrons in their covalent bonds. This association where they're attracted to one another, where I've drawn this arrow, that is a hydrogen bond. I should have got my pen because it's hard to write with this mouse. Keep clicking buttons on the side of it. But yeah, so that is a hydrogen bond. And that association dictates pretty much every molecule in biology. And I can't wait. I know this is so nerdy, guys. I know it is. I can't wait to share with you guys why this works the way that it works and how it affects so much of life and why it affects life. It's pretty incredible. And it's exciting once you start to understand why at the actual atomic level, things are the way that they are. Why, somebody has a certain eye color versus a certain hair color, or why my voice might be lower than your girlfriend's voice, or anything like that. Why I'm only 5'4", and why is my husband only 5'4"? And I don't know. So all that stuff is determined by your genes, but guess what? Your genes are made up of atoms. And the reason that those atoms are associating with one another the way that they are and forming the double helix of the DNA, all of it is the hydrogen bond. Isn't that crazy? It's just that, and it's all controlled by, the different molecules that can be affecting that association between the positives and the negatives. It's not always oxygen, right? It can be other molecules as well. And then whether or not some things can even interact with water or want to at all. We know how, like when you put oil into water, it does not want to mix. Yeah, we call that hydrophobic. That's water fearing. So they don't mix well because of their lack of a charge. That's really all it is. Carbons and hydrogens repeating over and over again. They don't really have a charge associated with this, right? When I drew this for you guys, there's not really a charge associated with either of these. Nobody's hogging any electrons or anything. It's just sort of bland, sort of neutral. So neutral things don't mix well with the charged things either. So that determines the shape of a lot of molecules in biology, and it is super exciting. You should be more excited than you are. I know you're not. You can already see it through this camera. So anyway, so those are our bonds, our building blocks, if you will. Those three bonds there probably give you more information than almost any of the rest of it. Okay. Next, organic molecules. Organic molecules, anything with a carbon-hydrogen backbone. So it has to be carbon and hydrogen, not carbon with oxygen or phosphorus with hydrogen, none of that. Carbon-hydrogen backbone. So when we talk about something like sodium bicarb, which is, we have sodium and we have this. All right. I'm sorry. Anyways, right. Anyways, so we have carbonate involves this too. We have carbonate involved here. That doesn't have any hydrogens in it. That is not an organic molecule. That's another thing that I want to reiterate. CO anything, that's not going to be, it has to have the hydrogen carbon backbone to it. Okay, keep that. So that's organic molecules, carbon and hydrogen, carbon and hydrogen. All right, the most important solvent for life is water. We've already talked about that, and the hydrogen bonds that are involved in it and all of the charges of those atoms that are involved in that, right? Very important. That is extremely important, which even leads us into this concept of pH. pH is a measure of the hydrogen ion concentration in a solution. So sometimes, a lot of times whenever we have water or solutions that are mixed with water, we don't just have H2O and like the nice little, pockets of, two H's attached to an O. Sometimes these attachments can become so strong or whatever that we can actually pull apart those atoms. We can have hydrogen ions, which show up as this. They don't have their electron anymore. Why? Because oxygen freaking stole it, because it does that, right? So we'll have that, and then we'll have what we call these OH groups, where we have the negative charge from that oxygen because we stole it from that other hydrogen. So this stole its electron from them, and now they're dissociated, right? Almost like ions, right? So in water, this can happen spontaneously. It's sort of like Schrodinger's cat. Okay, so if you know, like Schrodinger's cat, the story is that anytime that the box is closed, the cat can be alive and dead simultaneously. And once you open it, It has to be one or the other, basically. So it's sort of like how water works. So that's a real watered down version of it, but yeah, pun intended. But basically, so you'll have the normal water molecule H2O that we're thinking of, and then these ions of OH and H. So H, this is called a proton. It doesn't have any electron associated with it. So the only thing we care about is the proton, literally just proton, like proton, what we've been talking about with atoms. Okay. That's really all it is. It's just a proton. And it does have its neutron, but we're not getting into the neutron. The proton is what's important. So this is called a hydroxyl group. Hydro, going with the hydrogen, oxyl with the oxygen, hydroxyl group. Okay, or a hydroxyl ion whenever it has been disassociated and has a little negative charge and is doing its thing. Okay, that's important to understand for pH, because in plain old water, if all it is water in there, your H's and your OH's should be equal to one another, right? So the pH in that circumstance can go from 0 up to 14. If your H's and your OH's, your protons and your hydroxyls are equal to one another, the middle of that 0 to 14 is a 7. They equal out one another. Okay, so that's a neutral pH. That's like your ground there. Anything below 7 is going to be acid, and anything above 7 is going to be basic. So why? What does that mean? Well, the problem is this gets into math, and I friggin' hate it. I hate math, you guys. So, but we're going to get into it for a moment. pH, that value that you get, that 7 or whatever, think of it as corresponding to 10 to the negative something. So if it's 10 to the negative 7, and that is neutral, and now we have 10 to the negative 2, and we will call that 7, pH of 7, because we're just like, oh, that's the number we care about, is this one up here, the exponent, right? So that's a 7. So 10 to the negative 2, well, actually, if you look at it, 10 to the negative 2, and that is like that, right? There's 1 and 2. Okay. How many decimal points that you move it, right? To refresh your memory on these things. 10 to the negative 2 is more than 10 to the negative 7. You get that? So if I have 10 to the negative 2 concentration of hydrogen ions, is that more hydrogen ions than what we see in the 7 or less? It's more, right? So 10 to the negative 2 is more than 10 to the negative 7, which was neutral. That means we have more hydrogen ions, because this is a measure of the hydrogen ions, okay? That's why H. And that is a pH of 2. So do you see now how we've gotten a pH of 2, which is less than 7, and even though that number is less than 7, the actual concentration of hydrogen ions that we are referring to is going up. That's what that is actually referring to. So 10 to the negative 2. If it helps you to think about it that way, that's fine. If I'm talking about, oh, it had a pH of 10 to the negative, it had a pH of 10. Just say that, okay? Oh, we're talking about a solution that had a pH of 10. And 10 is higher than 7. So it should be basic, because that's like the watered down baby version of everything. To what you should be thinking when I say pH, instantly your mind should be thinking 10 to the negative, whatever that number is. Okay. So if it's 10 to the negative 10, we have less hydrogen ions than 10 to the negative 7. We have less hydrogen ions. That means the less hydrogen ions that are in the solution, the more hydroxyl there's going to be. And that means more basic. So think about it that way. It'll make more sense to you. And you do need to understand why these pHs are high and low and what it means and everything like that. That's all I'm going to go over with it for now. If you guys need for me to explain pH in any more detail than what I've gone over here, then please let me know, and I will try to take more time, so maybe in the review before the exam or something like that. So keep that in mind and bring it back with you if we need to talk about this again. So that's pH. So we've talked about water, and we've talked about all that. I wish I hadn't just erased all of this, but it is what it is. I'm going back now. No going back. All right. Yeah. So the biological macromolecules that we talk about in biochemistry, which is what we're going to be talking about in microbiology, are the carbohydrates, that's our sugars, our lipids, those are like fats, nucleic acids, that's like our DNA, and proteins. Proteins are the workhorses of everything. They're basically going to be fueling all of the reactions in our entire body, managing catabolism and metabolism and anabolism. All of those are pretty much the same thing. And all of these things that are going to go on in your body depend on the functions of proteins. Go figure. Okay, so those are the macromolecules we're going to get into. A lot of the shapes and functions of these guys are going to depend on their polarity. That literally just means if they have a positive or negative aspect to them, a charge, a dipole moment, or whatever you want to call it. If they have a positive or a negative to them or not. Okay. Things that interact well with water tend to have some sort of a charge to them. These are what we would call hydrophilic. Water, hydrophilic love. I used to live in Philadelphia, city of brotherly love, right? So we know what philic is. If it doesn't have any charge, like oils, all they are carbons and hydrogens. There's no charge associated with any of that crap. So that would be hydro, water, phobic, fearing. They're non-polar. Non-polar just means they don't got a charge. Okay, so that's what we've basically broken that down into. So what's this? Amphipathic. What does that mean? Amphipathic molecules have both going on. So they might have a charged end that might have, like, I don't know, a phosphate group, and phosphate has phosphorus, which is the P, and some oxygens associated with it. Usually 4 oxygens. Now, I've told you about how much hogs of electrons oxygens are. And they tend to steal and then become negative charge, right? So I just popped 4 of these guys on there. So now you know, without me having to tell you because you're smart, that this is going to have a strong negative charge. And it looks stupid the way I drew it, but yeah, strong negative charge associated with that one, right? Okay, but we can put this guy onto one of those carbon hydrogens. And so I will say that at every point that I put a little like notch, this would be a carbon, this would be a carbon, this is a carbon, and so on and so forth. You can keep drawing it like that and just know that there's a carbon at every single junction, okay? And they have their appropriate, hydrogens associated with it to fill out and be nice and whatever. So, but imagine this long, long chain that doesn't have any charge on it. This part would be hydrophobic, water fearing, right? That part of it would be. It doesn't want to associate with charge. This part, just at the head of it though, that is hydrophilic, water loving, right? So it's because it's got a charge. So that's amphipathic. It's got both a charged portion and the non-polar portion, the hydrophobic portion. And these molecules are very important in biology as well. Why? Well, we're going to talk a little bit about, when we get into talking about cells a little more, the lipid bilayer. And the lipid bilayer, this would be like the lipid portion, the hydrophobic portion would be this long little tail over here. And then this is the charged part, right? So these guys will, stick together essentially with their charged portions, and they'll be exposed to the outside where all the water is. And water's hanging out here. It's our solvent. It's biological solvent. So I don't mean it's not literal water, but whatever like the outside is. Okay, it's usually water-based, right? So yeah, hang in there. So we've got all these charged heads that will hang out with one another. We've got these negative charges. And then we've got these stupid tails that don't want to have anything to do with the water, but they will hang out with each other. Okay. So that makes sense. They're lining up and doing this. But what about like the inside of the cell is based out of water too? If we're talking about this being a cell, right? So this is out. and this is N, but both of them are based in a solvent that is water-based. Okay, so how does this work? Well, it's actually quite simple. We just have another layer of these guys. Let me just bear with me. It's forever on this. So they've got their little tails too, and their tails will stick up against these other tails. And so now they're completely sequestered away from the water. And this goes on, right? The head and the tails, and then eventually make a whole big old circle that encloses the whole cell. So now these guys that are afraid of water in this part in here, they don't have to be exposed to water. And now we've created a protective layer that is outside polar and can associate well with water. And inside non-polar doesn't want to have anything to do with water. So things can't even pass through it very easily, because how do you deal with that? How do you get through something like that where you've got both these situations going on? That is the lipid bilayer, and that is why your cells have membranes. So that is exactly what is going on there. And we'll talk more about that in another chapter, but it's extremely important, as I'm sure you can tell, that you understand why this works the way that it does, because otherwise you won't understand how we're getting things in and out of cells, and why we're talking about how to get things in and out of cells, and why pH is so important and everything like that. So moving on to the proteins, because we're not, again, going to talk about every single little thing in every little detail right here. You need to read the chapter for that. We'll listen to the very long lecture associated with the chapter for that. Let's go ahead and get rid of this chapter. Get rid of our lipid bilayer here. So, protein structure. Proteins are made up of building blocks called amino acids. This should sound familiar to you. An amino acid essentially has an amino group, which is a nitrogen NH2 group, right? And then the acid part is like carboxylic acid, so CO2 attached to it. And then in the middle, there's a carbon. So it looks sort of like this. Let's see how I draw it. NH2, CO2. Right. Basically, this is the idea. If we bump this up to NH3, put another one on there to make it happy, give it some charge here. So there's no hydrogen associated with this one, so it's got a negative charge. And then from this little carbon here, we'll have a hydrogen. Let's just round everything out real nice, shall we? Something we call an R group. Okay, so that R can be any number of things. It could be just an hydrogen, or it could be all these other little things that it could be. There are 20 different amino acids that could be made just by changing that R group that we know of. Okay, and basically, they could be polar or non-polar, or even have amphipathic qualities to them. And then based on whether they're polar or non-polar, or whether you have a whole bunch of polar or a whole bunch of non-polar all lined up and everything like that, of course, these atoms are going to react differently to one another, as well as to their environment that they're within, right? So if they're inside of a cell, inside of a water-based environment, and you've got a whole bunch of hydrophobic, they're going to want to fold in on each other and hide that hydrophobic water-fearing portion away from the water, because it doesn't want to have anything to do with it, right? That makes sense. So that's why amino acid sequence is so important. Basically, proteins are amino acids repeating over and over and over again. And then based on whatever that R group is going to determine whether it's polar or non-polar and what it can react with and everything like that, okay? That literal sequence of what amino acid, leucine, threonine, phenylalanine, tryptophan, all of those different amino acids that it could be, there's 20 of them. I'm not going to list them all. That is the primary structure of the protein. Just literally like the sequence of the amino acids that it has in it. Just basically the primary structure, this amino acid, this amino acid, this amino acid, this amino acid, all linked up together straight on through. Primary, that's what primary looks like, okay? Secondary structure, now we're having associations, like if I told you, we had a whole bunch of hydrophobic, water-fearing R groups together, they're going to want to hide in on one another. Or if they like hanging out with the water, maybe they want to associate more with the water in their area, and then the other area down further doesn't want to. So, based on all of that, literally based on all of that is how that protein is going to fold up and start creating its shape. The secondary structure is determined by those amino acid interactions with one another in that peptide, okay? That chain of amino acids that we've made, how they interact with one another and start forming their initial shapes. The first shape that we would talk about would be the alpha helix. It is just literally like a helix. This is the alpha helix. And then a beta sheet is usually drawn like this. It's an arrow. And they're basically just long stretches of non-folding area. So that's the beta sheet. You start seeing alpha helices and beta sheets in the secondary structure. All right, next is the tertiary. Things are getting a little bit more complicated. So instead of interacting with the guys that are immediately next to you, now we're like maybe way on down, further down the line, things that are across from you, interacting with them due to their charges or something like that. And one of the more common associations that are going to affect the tertiary structure are going to be disulfide bonds. That literally means that in that R group, if you had a sulfur group and across from it, way across from it, the same peptide, way further down is another sulfur group, they can form covalent bonds to one another. They're actually sharing their electrons now. So actually good, solid, stable, not hydrogen bonds associated with charge, but actually sharing electrons with like permanent bonds. So yeah, so that's how the tertiary structure is going to work. So associating across from those smaller structures that we've made, right? How those alpha helices are going to be associated with other alpha helices across the way, things like that. All right, so we will say disulfide bonds here, and then quaternary structure. So now we've folded up this whole mess based on the hydrogen bonds and the disulfide bonds and everything like that. And we've made a little like, I don't know, here's your peptide, that one single chain is bent up. So if your protein works with several like parts of these guys interacting with one another, and how they interact with one another through hydrogen bonds usually, or disulfide bonds even, that is your functioning protein. So we can have little peptides coming together and interacting with this blob and this blob and this blob and this blob, come together and now as one function as a protein. That's the quaternary structure. We can see here, as I'm explaining to you already in especially the first two, but especially if we're talking about the first three, and then later on the four, how important hydrogen bonds can be in all of this, right? The ability for hydrogen ions, the ability for hydrogen bonds to have an effect on these structures depend on the pH of the environment that they're in. So if our normal cells are like 7.2 to 7.4, if we bump that up where there's higher hydrogen concentrations, we could be interfering with how the protein would be interacting with other areas of the protein. If we bump the pH up or if we bump the pH down, make it more acidic or make it more basic, either of those can affect quite severely how those molecules within the protein are going to be associating with one another. And that is crucial to the function of the protein itself, and therefore the function of the cell, which is why pH is so important in biology. I hope that helped clarify some of the stuff that was in this chapter, and we can always review more whenever we get into the actual exam review. So bring some of those questions with you when we do start reviewing for that. And I will see you guys next time. That is it for chapter 2.